Hello students, welcome to NEET Tube JN classes, and in this today's class we are discussing about the concept, second PUC concept, biotechnology and its principles. First of all, what is this biotechnology? What is this biotechnology, and with this biotechnology what is the use, what is the purpose of this biotechnology? First thing is, in this biotechnology there is a presence of so many number of terms which includes, number one is preparation of recombinant DNA. The first thing is preparation of recombinant DNA. What is the meaning of this word recombinant DNA? Remember here, re means again, combination, combining. Yes, again we will combine the DNA. Yes, what sir? Whose DNA we will combine? We are going to combine the two organisms' DNA. Sir, which two organisms' DNA we have to combine, sir? Now remember here, for example, you take the organism is A and you take the organism B. Any organism, whatever the organism you want to take, you take that organism. First thing, the main thing is we have to prepare the recombinant DNA. Recombinant DNA means again we are combining the DNA. How many DNAs we are combining? Two organisms' DNAs we are combining. So let's take that one organism as A and another organism as B. Just remember, for example, this is the organism A. This is the organism A. This is the organism A. In this organism there is a presence of a cell. Cell definitely contain a nucleus. Nucleus contain chromosome. Chromosome is nothing but it is having the approximately 2.2 meters of DNA. From this DNA, we have to take a particular gene we have to take. Why? Because it contains a millions of genes. Millions of genes are present inside of the DNA. Millions in molecular basis of inheritance. In my lecture, molecular basis of inheritance, you comes to know that a DNA contains a number of genes. In the genes, we are very much particularly interested in one gene. Why we are interested in this one gene? Later, let's discuss. This gene, be careful, it is the cell. The cell contains a nucleus. A cell contains a nucleus. Nucleus contains a chromosome. Chromosome contains a DNA. Yeah, this is the long polymer. DNA is the long polymer. This is the DNA. In this DNA, a particular point is required for me, which is very, very interested in this point. So this point we call it as gene of interest. Yeah, this is gene of interest. We call it as gene of interest. Yeah, fine. This is gene of interest. This gene of interest, I want to transform into the another organism which I have taken it as a organism B. This is the organism B, and I have to transfer this gene of interest into the organism B. How can I transfer? Yes, it is not any human being or it is not anything. It is a gene which is very, very small, which we are not able to see. How can I transfer this gene of interest, a particular point of a gene, into the organism B? That is the main thing. For that transferring purpose, we require another DNA. That DNA is circular DNA. It is circular. So that DNA is circular DNA. This circular DNA we call it as vector. That circular DNA we call it as vector. Okay, what is vector? Means nothing but DNA, which we see in the prokaryotic organism, nothing but plasmid. In cell, the basic unit of you will get this circular DNA, nothing but there is a presence of circular DNA which is acting as a vector. Just remember, for example, this is the gene of interest. Now this gene of interest, directly I can't transfer this gene of interest from A to B. Why? Because it is a small gene, na? So because of that only I want one transporter. Someone should transport this gene of interest from gene A to gene B. For example, who is transporting? Okay, let's remember, it is transporting this gene of interest. I kept inside of a one bus. I taken a one bus. This is the bus. This gene of interest I am placing inside of this bus. This is the gene of interest that I place inside of the bus. Now this bus is transporting that gene of interest into the organism B's body. So now let's see, bus help in what? The bus help in the transport of that gene of interest. That bus helped in the transport of that gene of interest. So the bus is acting as vector. The bus is acting as vector. V-E-C-T-O-R, vector. Bus is acting as vector. Means what is vector? Something is transported by someone. For example, we have to transport from one place to another place. Okay, I want to move from Andhra Pradesh to, I want to move towards the, for example, you go for the Bihar. I want to move from Andhra Pradesh to Bihar. How can I go? I can go by plane, I can go by train, or I can go by bus, I can go by car. Means at that time I am transporting. Who is transporting me? That transporter is acting as vector. Vector means something that is carrying from one place to another place. In human health and disease, you comes to know that biological vector, mechanical vector. For example, malaria disease. Malaria disease is caused by this Plasmodium vivax. How this Plasmodium vivax is transported? That Plasmodium vivax is transported by the mosquito, which is known as the female Anopheles mosquito. That female Anopheles mosquito is transporting the Plasmodium vivax protozoa from one person to another person, from infected person to non-infected person. So mosquito is acting as what? Vector. Same like that only cockroach, housefly, they will transfer lot of diseases. So the organisms who are transporting from one place to another place, the transporter is acting as a vector. The best example. Now we want gene of interest. That gene of interest is present in the DNA. So we taken out the DNA and this is our gene of interest. This only particular gene of interest we are placing inside of the bus. Now the bus is acting as what? Vector, and it is transporting into the organism B. And after transporting into organism B, now the gene of interest is present in the organism A. This is the gene of interest of organism A, now which is present in the organism B. Now this gene of interest, what is happening? This gene of interest is going to replicate, doubling and increasing in a number. It will increase in a number. For what purpose we have to increase all this, sir? For what purpose we have to increase all this? Listen here. For example, the lot of human beings nowadays, the entire India is suffering from the disease which is known as the diabetes insipidus. Diabetes mellitus. Nothing but sugar. The cause of sugar is what? Because of lack of a protein, a peptide which is known as insulin. The insulin protein is very, very important. Then proteins are formed by what? Protein is formed by the translation of mRNA. mRNA formed by the transcription of DNA. So DNA contain a gene. For example, you consider this is the gene. This gene will undergo the replication. After replication, it will undergo the transcription. After transcription, it is undergoing the translation and finally synthesizing the protein in our body. What is that protein? Means insulin. So because of presence of insulin in this person's body, he is not suffering from the diabetes. So now you remember, we want to prepare the insulin then. Why? Because lot of diabetic patients are there. They have to use the insulin shot, insulin injection or insulin sugar tablet they have to take. How they are preparing? How they are preparing this insulin injection? Weekly once or twice they will take. How they are preparing the sugar tablet? Yes, this is all the sugar tablets are preparing because of this biotechnology only, by the processor. So a person's DNA, a small piece, a gene of interest is extracted from a person A. Now this gene of interest will code for the protein. Which protein? Insulin. Carefully this gene of interest is placed inside of the vector. And now this vector, with the help of this vector, this gene of interest we are sending into the person B. After sending into the organism B, we are going to increase its number. Majorly, what are those organisms? B organisms are bacteria. Majorly we are using the bacteria, and with this bacteria what is happening, bacteria is going to get divide very fastly and one become two at the time. Actually here one bacterial cell which contain only one gene of interest, if one became two gene of interest, two became four gene of interest, four became 8. In this manner multiplying, increasing in a number, and we can prepare so many number of gene of interest, yeah. This is the main theme here. In this biotechnology, the main theme is this one only. Now let's see how it is going to happen. Yeah, listen. DNA, which is a very long polymer, which is the present inside of this cell. It's not easy to do all this. I said orally very fastly, but it's not an easy to prepare this recombinant DNA. Now let's see. First of all, how to prepare this recombinant DNA? Preparation of recombinant DNA. Yes. How we will prepare this recombinant DNA? Number one, the first step in the preparation of recombinant DNA is isolation of DNA. What is the meaning of isolation? Isolation means taking out. Isolation of DNA. Whose DNA? For example, you have taken the DNA of organism A. Any organism you can take. Animal cell, plant cell, bacterial cell or fungal cell, any cell you want you can take. Whatever DNA you want you can take. Isolation of DNA. Yes, DNA is a long polymer. After isolation of DNA, we want only gene of interest. Means we don't want remaining all the parts. So what we have to do? We have to cut the DNA. So second part is cutting the fragmentation of DNA. Yes, how can we cut this DNA? Which is a very long thread polymer which we can't able to see. Why? Because DNA means it is present. Cell only we are unable to see. Then where is DNA? Cell, nucleus inside of chromosome. That chromosomal material is made up of this DNA. So fragmentation means cutting of DNA is only possible because of molecular scissors. We call them as. Why? Because these are cutting like what? Scissors. They are cutting the molecule. Which molecule they are cutting? They are cutting the DNA molecule. Because of those scissors, we call them as molecular scissors. So, with this molecular scissors, for example, a long DNA, with this molecular scissors, we are cutting that DNA into number of fragments. See, this is the fragment of DNA, and this is another fragment of DNA, and another fragment of DNA. So, we make the fragments of DNA. So, it is fragmentation of DNA. It is only possible by using the molecular scissors or a restriction endonuclease enzyme. I will tell what is this restriction endonuclease enzyme. And after making the fragmentation of DNA, once again, all the fragments we don't want. We got so many number of fragments, na? All the fragments we don't want. We want only the one fragment. The fragment which you want, that fragment is you require. So we want only that one. So we have to separate all those. The third step is the separation of DNA fragments. Yes, how can we separate? It can separate by using the gel. And also by using the electric field. So it is called gel electrophoresis. The process is called what? Gel electrophoresis. Separation of DNA fragment. Which DNA fragment? Which we require. So such DNA fragment we call it as desired DNA fragment. Desired means here what we require. Which is nothing but the gene of interest. We have to separate that one. By the process of what? Gel electrophoresis. By the process of gel. Okay, fine. After getting the desired DNA fragment, yeah. After that, fourth step. The fourth step is ligation. What is the meaning of ligation? Ligation means joining. Ligation means what? Joining. Yes, sir. Joining of this desired DNA fragment, nothing but gene of interest, with whom? Vector. With vector. With vector. For example, this is the vector, nothing but the circular DNA. This is the vector, nothing but circular DNA. We have to join this desired DNA fragment, nothing but gene of interest, like this. Like this we have to join. Like this we have to join. Whenever you joined these, see, here there is a presence of vector DNA, and this is the human's DNA, what you taken. For example, human cell you have taken. So this is the human's DNA. So two DNAs are present. This is the vector DNA, nothing but bacterial DNA or plasmid, and this is our gene of interest. Our gene of interest. So what happened? We cutted the DNA and we joined the DNA. So this we call it as recombined DNA. Or recombinant DNA. This is so recombinant DNA contains how many organisms? DNA? Two organisms. Number one is vector DNA, which is circular, and along with vector DNA there is a presence of our gene of interest. Now this is the recombinant DNA. And what we have to do? This recombinant DNA, what we have to do? Means we have to transfer this recombinant DNA into the organism B. Organism B. What organism you selected into that organism body? We have to transfer this recombinant DNA. And after transferring, you have to take care of that organism. Culturing that organism. Taking care of that organism. Culturing. Multiplying. What is the meaning of culturing? Aquaculture. Aquaculture means what? For example, increasing, culturing, cultivating the aquatic organisms. That is aquaculturing means what? Increasing. So, culturing of this organism. This organism, why we have to culture? Why? Because this organism contains the recombinant DNA. In this recombinant DNA, what is there? Gene of interest. For that means multiplying. 1 become 2, 2 become 4. And after that, from that, isolating the product. Isolating the products. This is the gene of interest of what? Insulin gene. So this gene of interest will give the protein. What protein? Insulin. We have to isolate that insulin product. This is the whole procedure in this chapter that you have to learn. In this chapter, these are the 5 important points. Recombinant. How to prepare this recombinant DNA? All this. First, isolation of DNA. Next, fragmentation of DNA. Next, separation of the desired DNA fragment. First 3 points let's discuss. Let's discuss first 3 points in this now. Now let's discuss first 3 points. Isolation of DNA. Yeah, how isolation will happen? And remember, which organisms DNA we require? Which organisms DNA we require means any organism you can take. For example, I want the organism. For example, I have taken this is the animal cell. It is the animal cell. Animal cell means cell definitely contains what? Cell definitely contains nucleus, and in that nucleus there is a presence of DNA. Now this is the DNA, which is a very long. Which is very long. This is the what cell? This is the animal cell. It is animal cell. Fine. Next, I have taken the one more cell. What is that one more cell? This is the one more organism cell. And cell definitely contains nucleus. Nucleus contains the molecule. What is a molecule? DNA molecule. And this is whose cell? Plant cell. It is plant cell. And after that, you might be knowing that it is cell membrane. And in a plant, there is a presence of wall. What wall? Cell wall, like this. There is a presence of cell wall, which is very rigid. Cell wall is present, and it is not going to bend. It is not making the cell. And the cell wall is made up of what? Yes, the cell wall is made up of cellulose. Cellulose is a what? Sugar. Cellulose is what? Sugar. Ose. If any word is ending with ose, it is a sugar. Glucose, sucrose, lactose, galactose, fructose, maltose, cellulose. In this manner, any word, if it is ending with ose, that is telling that it is made up of a sugar. Now it is what? Cellulose. Cell wall. Plant cell wall is made up of cellulose, nothing but sugar. Another cell also you can take. What is another cell that I want to take is fungi cell, mycota cell, fungi. This is the fungal cell. Fungal cell also definitely contains what? Nucleus. Nucleus contains a molecule. What molecule? DNA molecule. This is the cell fungi. fungal cell. And the fungal cell also having the wall. What wall it is also having? Cell wall. Yeah, the cell is covered with the wall which is known as the cell wall. This is the cell wall. And the cell wall in fungi is made up of what? Chitin. Chitin. It is made up of chitin. The cell wall in fungi is made up of water? Chitin. Yes. Which is a very, very rigid. And next one. You can also take the bacterial cell. That is our wish. You can also take the bacterial cell. This is the bacterial cell. And the bacterial cell also having the DNA, where the DNA is naked. Nuclear membrane is absent here. Nuclear membrane present. Nuclear membrane present. Nuclear membrane present. Prokaryote, and it is, sorry, these are eukaryotes and this is prokaryote. Nuclear membrane is absent, prokaryote. Nuclear membrane is present, eukaryote. All these three are eukaryote and it is a prokaryote. Now the even bacterial cell is also covered with the cell wall like this. There is a presence of cell wall, peptidoglycan, which is made up of peptidoglycan. Yes, this is what cell? Bacterial cell. Bacterial cell. Whose cell's DNA you want? Why? Because what is the first step? The first step is isolation of DNA. Isolation of DNA. This is the first step in the preparation of recombinant DNA. So, whose cell's DNA you want to take? Whether animal cell, plant cell or fungal cell or bacterial cell. That is your wish. Yes, now you remember. This animal cell, to take this desired DNA fragment, let's take one test tube. One test tube you have taken. This is the test tube. Inside of this test tube, you have placed the cell. What cell? And animal cell you have placed. This is the animal cell, which contains a long polymer that is nothing but DNA. We have to take the DNA. How? It is not as easy. Why? Because it is having the cell membrane. Cell membrane is made up of what? The cell membrane is made up of proteins. Cell membrane is made up of proteins. Not only proteins, the cell membrane is also made up of lipids. And lipids are arranged in bilayer. Lipids are arranged in bilayer. Cell, the basic unit of life, you will get all this. The cell membrane is made up of protein and as well as the lipids. And along with, there is a presence of so many number of RNA molecules inside of the cell. rRNA, tRNA, mRNA, all the RNA molecules. Different type of RNA molecules are present. There is a presence. And along with that, there is also presence of so many number of proteins are also present in the cytoplasm. So many number of proteins also might be present inside of the cytoplasm. All these are the proteins. Yeah, we don't want anything. We don't want proteins. We don't want lipids. We don't want RNA. We want only the DNA. We want only the DNA. How can we take this DNA? It is not a chocolate, na? Opening the wrapper and taking the chocolate. It's not an easy. So because of that only, we have to treat this animal cell with enzymes. Enzyme. And most of the enzymes are almost protein and almost enzyme names are given based upon their function. Based upon their function. For example, I am the one enzyme and I can break the proteins. I will break the proteins. I will remove the protein. So my name is protease. Protease. Why? Because enzyme name ends with ase and you are taking out the proteins, so it is a protease enzyme. And next there is another enzyme which is breaking the lipids. So what is the enzyme name? Lipase. Lipase enzyme which break the lipids. And there is also presence of RNA. RNA is also a polymer. We don't want RNA. How can I break? There is one enzyme. Yes, I am the enzyme which I can break RNA. So the enzyme name is RNase. The enzyme name is RNase. In this manner, what is happening? Cell membrane is made up of proteins and lipids are those enzymes broken all those protein cell membrane. Complete cell membrane is destroyed. Now these are the particles of all the black dot is indicating the broken parts of cell membrane. Broken part of cell membrane. And there is also presence of lipids. Okay, let's represent. All these lipids are broken down and all these green dots is indicating the broken parts of lipids. And there is also breaking down of RNA is also happened. So all these are the broken parts of RNA. All these blue dots. In this manner there is a presence of broken dot of RNA. Now let's see. DNA is mixed with so many number of broken particles. DNA is mixed. Yeah, fine. Like one thread is present and all waste particles are present. We don't want all those waste particles. We want only the DNA. So how can we take out this DNA? Why? Because all are mixed now. So for separation of this DNA from all this waste material like which are broken of protein parts, lipid parts and RNA part, to separate this DNA, we have to add the chilled ethanol. We have to add the chilled ethanol, means C2H5OH. Okay, add that. We have to add the chilled ethanol. Whenever you added the chilled ethanol, this is important. What is we are adding? Chilled ethanol. Whenever you added the chilled ethanol, at that time, the DNA which is precipitated down. So at that time, what is going to happen means the DNA is precipitated down. Yes, DNA which is a long polymer. Yes, the DNA is precipitated in the bottom. The DNA is precipitated in the bottom. All the broken down of particles like proteins, lipid broken down particles and as well as RNA broken down particles, all are present on the top. So what happened? The DNA and as well as all the waste materials are separated. And this DNA, we have to take it outside by keeping one spoon. By keeping one spoon, we have to take that DNA outside like this. We have to take it. This process of taking out of DNA is called spooling. S-P-O-O-L-I-N-G. Spooling. What is spooling? The process of taking out of DNA is called spooling. So in this manner, what we did, isolation of DNA. Finally, I have taken the DNA out of the animal cell. Out of the animal cell. In this manner, we have to remove isolation of DNA. Whose DNA I have taken? Animal cells DNA. So if you want to take the plant cells DNA, first we have to break the cell wall. The cell wall is made up of what? Cellulose. So to break this cellulose, we have to add the enzyme cellulase. We have to add the enzyme cellulase. And remaining everything is same. If cellulose is broken down, next plasma membrane, protease, lipid, lipase, RNase, etc. If you want to take the fungal cells DNA, at the time adding the fungal cell inside of the test tube. So cell wall is present chitin. The chitin cell wall is broken down by the chitinase. It is broken down by the chitinase. And after that plasma membrane etc. everything is same. Yes, isolation of DNA by the process of spooling. And next one is what? Bacterial cell. If you want the bacterial cells DNA, same treatment, add the test tube, but cell wall is present. That cell wall is digested by the lysozyme. Enzyme is what? Lysozyme. So, in these three animals, you have to add one extra enzyme. Why? Because these three cells contain cell wall. Cellulose. If you add plant cell at that time to break that cell wall, cellulase. If you add fungal cell to break that chitinase. If you add the bacterial cell, you have to add the lysozyme. For animal cell, there is no cell wall, so there is no need of adding of. Remaining all protease, lipase, RNase, etc. Everything is same. Process of taking of DNA, spooling. And the third one is what is adding for the separation of DNA? Chilled ethanol. The chilled ethanol is added for separation of DNA, and after separation you are taking out with the spoon, small threads. That process is called spooling, and finally you taken out the DNA. So isolation of DNA is happened. First part. Isolation of DNA. In this isolation of DNA, I don't want this entire DNA. I want only a small piece. I am only interested in the one small piece. That small piece I can call it as gene of interest. This small piece I want. So remaining all I don't want. So we have to break this. We have to cut the DNA with molecular scissors. So second step is fragmentation of DNA. So cutting the DNA into number of fragments. That is the second step. Only 2 points are required. The process of taking out of DNA is called what? Spooling. What is the ethanol that is added? Chilled ethanol, which is a very, very cooled ethanol, alcohol, nothing but is added. Now fragmentation of DNA. What is this fragmentation of DNA? This fragmentation of DNA is fragmentation of DNA. Isolated DNA. For example, DNA is a very, very long polymer. Yeah, you represent this is the DNA, one strand, and you represent the another strand, and this is the another strand of the DNA. Another strand of the DNA. One strand having the 3 prime, opposite is having the 5 prime, and here we having the 5 prime, and here we having the 3 prime. This you might be knowing. So in this I have a one particular point which is where I am interested. That is the gene of interest. This is my gene of interest. Blue is my gene of interest. For that purpose I have to add a molecular scissor. Yeah, this is the molecular scissors. These are the molecular. With these molecular scissors are called restriction endonucleases. Before going into that, first of all, it is the DNA. DNA means what? Na means what? Nucleic acid. So it is a nucleic acid. It is a nucleic acid, right? DNA means what? Deoxyribose nucleic acid. So it is a nucleic acid, one nucleic acid. Fine. Now this nucleic acid is cutting. Who will cut? One enzyme will cut. One enzyme will cut this nucleic acid. So that enzyme, I am the enzyme who I will cut this nucleic acid. So that enzyme name is what? Nuclease. Why? Because enzymes are given the name. Most of the enzymes are given the name based upon their functional, protein, protease, cellulose, cellulase, chitin, chitinase, lipids, lipase. In this manner. So this is the enzyme which is cutting the nucleic acid. So the enzyme name is nuclease. If this nuclease enzyme, it is of two types here. It is of two types. The nuclease enzyme is of two types. If it is cutting the DNA from corner to center, from edge to the center, like this, from in this direction it is cutting. In this direction it is cutting. From corner to center. In this direction it is cutting. From corner to center. At that time that nuclease is called exonuclease. Exo means what? Outside. Who is cutting? Enzyme. What is enzyme name? Nuclease. So it is cutting from outside to inside. So it is exonuclease. Not like that. I am the enzyme. I will cut the DNA in a different direction. Means how you will cut? I can cut the DNA from center to the out. Like this I can cut. From center to outside I will cut. At that time the enzyme name is endonuclease. Both are nuclease. Nuclease means what? Which will cut the nucleic acid. Nuclease means what? Which can cut the nucleic acid. Which nucleic acid? DNA or RNA we can say. DNA. So if it is cutting from out to inside, exonuclease. If it is cutting to into out, it is endonuclease. That is the only difference. So most of the time we will use the restriction endonuclease. What is the meaning of this restriction, sir? Restriction means order. Hey, I am restricting you. You have to sit down here only. You have to eat this only. I am restricting you. So this restriction endonuclease, this nuclease having one particular site. There is a one particular site will be there. In that particular site only it will cut the DNA. And nothing like that. Only at a particular site they will recognize one site. This is my site. I will cut here. And they will recognize site means checking. Oh, this is my site. Cutting. And checking. And recognize cutting. And checking. Recognizing cutting. In this manner, different type of endonucleases are present. And different endonucleases have different recognizing site. What are the sites? Means these are DNA means adenine, guanine, cytosine, guanine, adenine, thymine. In this manner, it will be present. Nitrogen base pairs. So by recognizing those nitrogen base pairs, they will find one recognizing site. They will search. This is my site. Cut. This is my site. Cut. This is not my site. I won't cut here. This is not my site. I won't cut. This is my site. It will. In this manner, it is cutting the DNA into a number of fragments. It is cutting the DNA into number of fragments. At that time, what the shape you are getting? Yes. At that time, see how the DNA is broken down into number of fragments. Broken down. In this broken down fragment, I want only one fragment I want. Yes, this is the fragment I want. So this is called desired DNA fragment. This is called desired DNA fragment. Yeah, how can I get this desired DNA fragment? How can I take this? It's not any marble, or it's not any chalk piece to take, etc. No, this is mine. This chalk piece is mine. This chocolate is not. This is the gene of interest. How can I take from this all this DNA fragment? So for that purpose only, there is a presence of separation. Third step. Yes. So many number of DNA fragments are present. Why? Because restriction endonuclease enzyme cutted the DNA into so many number of fragments, and all those fragments are together. In this fragment, I want only one fragment. So how can I take that one fragment? Collect all the fragment. Collected, keep aside. Collected, keep aside. Next, third event. Third event. What is the third event? What is the third event? In my hand, so many number of fragments are present. Whose fragments? DNA fragments are present. In this DNA fragment, I want only one fragment. So I want to separate. Separate. From this separated DNA fragment, I will take only one DNA fragment, that what I required, which is acting as a gene of interest. Now let's see, for that purpose, heading, a process we have to do for that separation purpose. So what we are doing? Separation of desired DNA fragment. Why? Because we got so many number of fragments, na? So one fragment, so we have to separate all the fragments. How can we separate? We can separate these DNA fragments by using a one gel. What gel? Agarose gel. Which is a liquid substances. This agarose gel is a polymer. P-O-L-Y. Poly. M-E-R. Mer means unit. Poly means many. Means it is made up of many units. Wall is a polymer. What is a monomer for wall? Brick. Wall is a polymer, right? It is made up of many units. No? What is a monomer for the wall? Brick. Now same like that only. Thus agarose gel is a polymer. Yes, we have to prepare it, sir. No, it will naturally grow. Naturally grow in the ocean, in the plants. Yes, so this is the which polymer? Natural polymer. It is a natural polymer. So extracting from where, sir? Seaweed. A plant which is known as the seaweed. Weed means unwanted plant, and that unwanted plant is present in the sea. So it is a seaweed. So gel. What is the gel name? Agarose gel. From where you are taking this agarose gel? First of all, agarose gel polymer or monomer? Polymer. Yes, what polymer? Artificial polymer or natural? Natural polymer. Naturally you will get from the plant. Which plant? Seaweed. Weeds which are present. Okay, this agarose gel, by using this gel, along with gel, we also use the electric field. And this gel having the pores. So this whole process is called gel electrophoresis. Yes, how you are separating the desired DNA, how you are separating the DNA fragments, all the DNA fragments, how you are separating? By using the gel electrophoresis technique. What is the gel? Agarose gel. Fine. Nowadays, there is a one bed for separation purpose. There is a presence of one bed. In this manner, there is a presence of a bed. This is the bed. Like this, there is a presence of bed. In this bed, we have to add the gel. What gel you adding? Agarose gel. Yes, just remember this. All this white color portion is the agarose gel. It is a gel. And here there is a presence of electric field. This electric field is minus, which is known as the cathode. And this on this bed, in this region, here there is a presence of another electric field. And this is positive. What is a positive? Anode. And here there is a presence of well. Wells will be present here. Wells means nothing but holes. Like this, there is a presence of openings will be present here. Like this, there is a presence of openings. Like this, there is a presence of openings will be present. Opening. Now we have DNA. These we call them as wells. Well are located in which region? Cathode region. Why, sir, they have to locate in cathode region? You will understand. Now these are the well. What we have to do? What is present here? Gel is present. Inside gel is liquid is present. Gel. Now let's see. We have the DNA fragment. Taken those DNA fragment in pocket now. Now we have taken all that. These are the DNA fragments. These are the DNA fragments. All these are DNA fragments. So now let's see, all the length of the DNA fragment is same? Never. Why? Because that restriction nuclease enzyme having different recognizing sites. So one fragment may be large, one fragment may be small, one fragment may be in medium, so different fragments. In this fragment, I want only one fragment, which is known as a gene of interest. Yeah. This is my fragment. You imagine this is my fragment. So what? I don't know. What fragment? I don't know. This is my gene of interest fragment. All the fragments are placed inside of this well. All the fragments we have to place inside of the well. Throw in the well. Throw in the well. And switch on the electric field. Switch on the electric field. What happens now? These DNA fragments are moving towards the positive charge. Why? Because the DNA fragments are a negatively charged molecule. DNA is what molecule? Negatively charged molecule. But negative always attracted towards where? Positive. Negative always attracted towards where? Positive. So DNA fragments are negatively charged molecules. Whenever we switch on the electric field, these DNA fragments are moving towards where? Anode. Anode is what? Positive. So the negatively charged molecules are moving towards positively charged molecule. Now let's see. All the DNA fragment having the different size. This is a small size. For example, this is the very small. Small size means it will run very fast, moving, and came here and settled in this point. In this manner, later one more you take, which is having the medium size. Yes, that medium size. Next one will be present here, and next one will be present here, and next one will be present here. See, according to their size, they are moving. According to their size, they are moving. So all are DNA fragments only. Small DNA, we placed all the fragments in the well. The small DNA fragments moved very far. Smaller the DNA fragment, far it will move. Far it is moving from cathode, and it is moving towards whom? Anode. If DNA fragment is very large, it will move only some distance because of its large size. Smaller the DNA size, farther it will move. Moving towards where? Anode. Why it is moving? DNA is negatively charged molecule. Now in this separated part, I have one DNA fragment. I want only one. So this is my desired DNA fragment. This I want. Now all the DNA fragments are present in where? Gel. We can't see this. We can't see the DNA. For that seeing purpose, once again, we have to add one chemical. Yes, add a chemical. We have to add one chemical. What is the chemical? The chemical is ethidium bromide. Ethidium bromide. Yes, the chemical we added. What is the chemical that we added? Ethidium bromide in the agarose gel. Yes, all this is the ethidium bromide. Okay, sir, when you add in the ethidium bromide, you can able to see the DNA fragments, No. After adding this ethidium bromide chemical, you have to pass the UV rays. Whenever you allow the UV rays, all the DNA fragments are appearing in the color. What color means? Bright orange color bands. Bright orange colored bands. Yeah. So, how can we see the DNA fragments? We can't see generally. To see the DNA fragments, we have to first add ethidium bromide. After adding ethidium bromide, we have to pass the UV rays light. Whenever you pass the UV rays light, at that time you will see the DNA fragments clearly in the color of what? Bright orange color. This color is very important. There, for isolation of DNA, we added the chilled ethanol. Here we added the ethidium bromide, and the DNA fragments are appearing in what color? Bright orange color. So in these, I have to take out this my gene of interest. So how can I take that one? Why? Because all this is what? Gel. In the gel it is present. So when you taking this desired DNA, gel also will come outside. Obviously, for example, take one coffee. In the coffee, some housefly is fallen. When housefly is fallen, you have to take that housefly. For that purpose, you take it a spoon. While you taking that housefly, you think that only housefly will come outside. No. Along with housefly, what will come in? Small amount of tea also will come outside. Now. Same like that. We have to take this gene of interest. So we have taken this process of taking out of all this is the desired DNA fragment. We taken out. Yes, we taken out. This is we taken out. We taken out. This is what? Gel. This is what? A gel which came along with the DNA, and in this gel only we have the desired DNA fragment. And this process of taking off desired DNA fragment is called elution. Elution. It is important. Elution. Don't get confused. Taking out of DNA is called spooling. Taking out of desired DNA fragment is called elution. Their chemical added is chilled ethanol. Here chemical added is ethidium bromide, and it is followed by the allowing the rays. What rays? UV rays, light. So in this manner, isolation of DNA completed. For different cells, we have used the different enzyme. After that, cutting the DNA by using the restriction and endonuclease enzyme. Nuclease enzyme two type: exonuclease, endonuclease. As of I told how to it is. After that, we want so many fragments you will get. In that fragment, we only want one fragment. That one fragment, how can we get? We have to separate all the fragment. Separation is done by the process gel electrophoresis. Gel name agarose gel, a natural polymer extracted from seaweed. And these fragments are separating according to their size. Small size, farther it will move. DNA is negatively charged molecule, so it is moving towards positive charge. Na? And after that, taking the desired DNA fragment. What the fragment you want? Only that one you are taking. This is called, process is called elution. For bright orange appearance purpose, added the ethidium bromide, and later we followed the UV rays. That's all. So while you're reading this chapter biotechnology, first thing that you have to read is processing of recombinant DNA, which is in the page number of 209 or 210 in your NCERT textbook. I think so, in your NCERT textbook. And after that, you have to come back and you have to search for this heading, separation of desired DNA fragments. There you will see this diagram. You find this diagram which is having the bright orange bands that you have to study. First, these two things you have to study, not the introduction. In biotechnology, you are not supposed to read the introduction. You won't understand anything. If you study the introduction, first thing is processing of recombinant DNA. For that purpose, few steps are there. What I written initially: isolation, fragmentation, separation, joining to ligation with the vector, that one. So this is the today's class regarding the biotechnology, and I will upload few more videos regarding this biotechnology, molecular base of inheritance, principles of inheritance. So I want your support, that I am asking you that please subscribe to the channel and like the channel, and as well as share these videos to your friends that more get benefited, and please give the comments that what you require and what doubts that you're having in any chapter of first PUC and second PUC, botany and as well as zoology. Thank you for all.